Hello everyone, and welcome to the Fossils and Phylogenies lab today. So we're going to be taking a lot of things that we've discussed in the past, because you all now hopefully are experts at sequence alignment, hopefully understand how to look at sequence data in the first place, and now you've even begun to construct some of your own phylogenetic groupings with our communicules from last week. Now we're going to take that information on how we build trees based on principles of maximum parsimony and molecular data and combine them so that way we can use lots and lots of data in order to build our trees. I'm sure last week y'all noticed the fact that using physiological traits, a lot is left to interpretation in terms of how different biologists view the physiology of the things that you're studying. How to interpret paint patterns, how to interpret structural differences. There is some wiggle room for how you interpret and design those character state matrices. Well, molecular data, as long as you're selecting good loci for it, leaves a lot less to interpretation because you are judging whether the bases are there or not and whether single nucleotide polymorphisms or SNPs are shared or not. And so here is our first alignment with our 3 different species, 1, 2, and 3, comparing to an outgroup. Now you'll notice that in the lab itself, there's a lot of extra letters in between. Everything in between each of these SNPs is conserved sequence. So, in terms of character state matrices, they're shared by all of them. So we don't need to use them for comparison. And that's going to be the case for the vast majority of the genome, right? If I'm comparing my genome to your genome, we're 99.99% similar to one another. And if I'm comparing myself to chimpanzees, I'm 99.9% similar to chimpanzees. There is a vast majority of the genome that's shared that is non-informative to help us with our groupings. So you'll notice that at least in this alignment and in future ones, and I encourage in the alignments that you draw as well, that you really focus on the sites that are polymorphic or variable. Now the next thing you're going to want to do when looking at these polymorphic sites is identify which ones are different from the outgroup. Which ones are establishing relationships between your species and separating them from our outgroups. So looking at our first site, we see that the outgroup had an A, species 1 and 2 have an A, and species 3 has a G. So based on this grouping, we see that 1, 2, and the outgroup should be together, and the third species, it's on its own. So if we were to do a small little phylogenetic tree from this, we could have a polytomy for the outgroup, 1 and 2, and then species 3 would have its tick mark to change SNP 1 to a G. And so in this way, you're going to be still mapping on your character state matrices SNP mutations, just like you would physiological changes. So let's go through a couple other of these and see how many are shared. Well, looking at our second site, it's shared across all 3. This allows us to group the outgroup separate from the species that we're studying. Site 3, we see groupings between the fact that the outgroup, species 1 and species 3, are the same. This separates species 2 from the rest of them. Next up, we've got a unique SNP to species 1. Next up, we've got a unique SNP to species 3. Next up, we've got a shared SNP between species 1 and 2. And you'll notice that I changed the emphasis in that word. When we've got something that is unique to one lineage, that's going to be a tick mark on a single branch. It's not going to be shared with anything else. When SNPs are not shared with anything else, they don't help us in our groupings. So yes, they're important to note because they can give you an idea of things like branch length, which again is not something we're going to emphasize in this class. But you're really looking to filter out the SNPs that are shared between different individuals, between different species, as a means of allowing you to group them together. So you'll notice that I'm going to put asterisks around the non-informative are the ones that are shared only by one species, and I'll be boxing those that help us distinguish multiple species together. So again, on SNP 7, we see groupings between 1, 2, and 3, different from the outgroup. Locus 8, we see groupings between species 1 and 2. And then locus 9, we see that species 1 has a unique SNP. So this is how I'd like you to generally be looking at your data. Identify the outgroup, which we assume to be the ancestral character for all of these loci. Now, it's an important discussion to have as to why we assume that it's ancestral. I want y'all to think about for a second here. If we're assuming that this is the ancestral state, and a vast majority of the sites are shared, why don't we assume that evolution happened independently on this branch? Why don't we assume that this C may have transitioned to a T and then back to a C? Think about the likelihood of mutation rates, and think about how big the genome is. All right, so hopefully you've had a little bit of time for food for thought there. While revisiting that topic about the likelihood of mutation, maybe y'all will remember that the likelihood of mutation at any given site is something like 3 times 10 to the minus 8. That's a lot of zeros. The likelihood of a mutation is incredibly low. Well, if the likelihood of mutation is incredibly low, and genome size is on the order of something like 3 billion base pairs, like a human genome, you need something that's incredibly rare to happen in the same spot when there are 3 billion opportunities for it to happen. This means that there is an incredibly low likelihood of mutations occurring in the same spot. We refer to this in population genetics as the infinite sites model, and it allows us to safely assume that no mutation is a remutation, that shared mutations occur because of shared ancestry, not because they happen twice. And that's absolutely critical. We're assuming that all of the mutations that are shared because of the fact that they share ancestry. Not independent events, but rather shared ancestry. And that's going to be critical to all of our molecular phylogenies. It's essential to understand in order to build your phylogenetic trees correctly for all these different traits, or all these different species, rather. So, we also want to highlight that because these genomes are very big and the mutation rates are very small, that a vast majority of these are conserved regions. And you want to select regions in kind of a Goldilocks situation. Again, you don't want regions that are hypervariable, and you don't want regions that are hyper-conserved. Because you can imagine that things like proteins in your mitochondria, well, they might be similar between me and you as 2 different humans, but also they can be exactly the same in my dog, also as a mammal. Things that are those maintenance genes, stuff that everything's going to have, are usually your highly conserved regions. And you want to make sure that you're not selecting something that can't change, nor do you want to select something that changes way too frequently. This does require a little bit of finessing and exploration, but for the purposes of our labs, I'll just be giving them to you. But I do want to plant that seed for those of you going into phylogenetics. Now, in terms of building the tree from this, what we're going to be doing is seeing where and how many traits are shared. So the first thing I'm going to do is go ahead and ignore all of our polymorphisms that occur in a single species. Then we're going to want to see how many mutations are shared between different species. Well, we see that site 2 and site 7 are shared across all 3, differentiating them from their outgroup. But in comparing 1 and 2 and 1 and 3, we see that 1 and 2 share more sites. They share this one, this one, this one, and this one. So there's a pairwise comparison index of 4 sites in common, whereas 1 and 3 share this one and this one, or a pairwise index of 2. This allows us to say that in the phylogenetic tree, looking at the outgroup, that species 1 and 2 share more recent common ancestry than 1 and 2 does with 3. And so then if we're placing these SNPs along the phylogeny, you can say that SNP 2 and SNP 7, SNP 2 and SNP 7 provide you 2 tick marks in the common ancestor between 3, 1, and 2, and that SNP 6 and SNP 8 are shared between just 1 and 2. And then you have a whole bunch that occur on each individual branch. SNP 1 is specific to group 3, SNP 3 is specific to group 2, SNP 4 is specific to group 1, and so on and so forth. And in this way, you can map along all of your SNPs along your phylogeny, paying very close attention to those that are allowing you to group things. With this information, I want you all to take a look at your next group of 4 species. We're looking at our group, species 1, 2, 3, and 4. Take some time to do the similar comparisons to identify pairwise differences. And I'll see y'all when we draw the phylogenetic tree on the board in a few minutes. Good luck. All right, so here is my completed tree. Hopefully you took time, paused the video, and did something like this yourself. Now looking at our 4 species, there's a couple things that should stick out to you. First and foremost, if you look at the alignment between species 4 and the outgroup, you'll notice that species 4 only has one difference, that second SNP, that change from an A in the outgroup to a G in the 4 different species. That's the only difference between these 2. That gives you a safe bet that species 4 is very, very closely related to our outgroup. Now, looking at the rest of our species here, species 1, 2, and 3, you want to again do those pairwise comparisons to see which ones are closely related to one another and which ones are closely related to the outgroup. Always doing these pairwise comparisons to allow clustering within groups as well as referencing to the outgroup. Now you'll notice that there are 2 sites that are shared by all of these, SNPs 6 and 9 if I did my numbering correctly, and that all 1, 2, and 3 inherit those sites. But then that 1 has a couple of singletons. These are unique to it, SNPs 5 and 7. But that SNPs 2 and 3 share a whopping 4 sites. Sites 1, 3, 8, and 10. Those are the alleles that are colored in, only on sites 2 and 3. So you notice that there are 4 of them. That should really drive home the point that species 2 and 3 share a most recent common ancestor, because they're inheriting so many of these shared mutations. Now you again have a singleton mutation in 3, 2 doesn't have any specific to it, 4 doesn't have anything specific to it, and therein lies the phylogenetic tree from that alignment in your lab. Now for your next exercise, we're going to reverse engineer this problem. Rather than giving you guys an alignment and asking you to draw a tree, I'm going to give you a tree and ask you to draw an alignment between several different species. So we're looking at an outgroup of zebrafish. I'm just going to use the first letter of these animals' names in order to indicate what species they are. Xenopus, or the frog, is next out. Then a chicken, the living descendants of dinosaurs. And then, sister to one another, are mice and humans. And your task is to design an alignment completely made up that will give you a tree that recapitulates this relationship, where human and mice are sister to one another, chickens are next out, frogs are next out, and zebrafish are our basal or outgroup species. There's infinite possibilities for this, but I want to show you at least the simplest process of doing this. you can design any site, so we'll call it snip 1, and we'll say that the zebrafish has an A at this site. Now, in order to group frog, chickens, mice, and humans together, zebrafish needs to be different from these 4. So we can go ahead and say that frogs inherit a G, chickens inherit a G, mice inherit a G, and humans inherit a G. This snip 1 therefore groups together our 4 species. Now next up, if you want zebrafish and frogs to group together at site 2, what you've got to do is a snip that is shared between the 2 of them. Any random letter will do. They share a T and a T. And then you want this to group chickens, mice, and humans together. So we'll go ahead and say that these inherit a C. Are you seeing a pattern here? In order to establish these groups, basically you can design a structured problem where the next site, if we want to group together mice and humans, you need zebrafish, frogs, and chickens to share an allele. Let's give them all a G. Let's say that mice and humans inherit a C. So what you should be noticing is that these shared snips that are different from our outgroup allow us to establish a hierarchy of a relationship. What you want to be doing, therefore, and thinking about when you're looking at these major trees, where I don't give you a structured problem like this, what's actually happening in reality is we're shuffling these around. They're not going to be in the same order. They're going to be shifted around. In a fake example, you can design something really neat and simple. In a more complex example, you're going to see a lot of shuffling. So I want you all to take some time, maybe design a tree that you can exchange with a friend, and see if you can build the same, or share an alignment that you can exchange with a friend, and see if your friend builds the same alignment, builds the same tree from that alignment. Next up, I'll see y'all over at our fossil cabinet to talk about some fossilized species. All right, well now that we can move on from some of our alignments, let's take a look at some of the biodiversity that once existed on planet Earth. So we've got a couple of different types of fossils here. Here in our cabinet, you'll notice actual fossils that are fossilized rock. And then you've got a couple of fossil peels that I'll keep back in the back room. So this is the first thing that I ask y'all to take a look at. Fossil peels are sections of what are called coal balls. We're able to take organic material bound to a cellulose piece of paper that sticks to it, and you're able to take thin sections of a rock off at a time. This allows you to look at these structures under the microscope. Now, I'd encourage you all to take a look at some of these other structures under the dissecting microscope. But right here, we have a very, very large structure that's visible with the naked eye. Here, you see the fiddlehead of a fern, the looping structure on some of the ferns that you see in the greenhouse today. These are the way in which their leaves grow. This plant was in a bog something like 300 plus million years ago, died, got buried in the bog, and was around during a time where fungi were not digesting it. This was the Carboniferous. That stayed in the ground and was dug up by us and placed on a fossil peel for students to look at forevermore. A lot of the same happened with some of our fossils in the fossil cabinet. For this exercise, I want y'all to take a look at all these fossils, but in particular, I'll label 4 that are really unique. Fossil A from Ginkgo phytopthys. I want y'all to notice the patterns in leaf venation here and think about what other species, maybe sharing a name, like ginkgo, is still alive today. Fossil B is our lepidodendron species. This looks like dragon scale, but it's a tree. This is the bark of a tree that's actually related to one of the plants in the aquarium, our Isoides. So give this a Google, Lepidodendrales, or lepidodendron, and find it on Wikipedia to see what it's related to today and how its bark actually looks like dragon scale. For fossil C, we'll go down to here, the Stylocalamides, one of the coolest names, I think, Calamides. spelled like calamities, kind of. It is a relative of horsetails or equisetum. You can see some of the banding patterns here and some of those vertical venation patterns that you'll see in horsetails today. The difference was that this would have been a tree trunk that would have been 2 feet wide. Horsetails are not nearly as big today. And our last fossil here is of one of the many, many, many ancient ferns. One of the many ancient ferns. So we've got a couple of different ferns and a couple of other selaginellas. You'll see another fern actually up here. You'll see a lot of similarities in the structure of these plants. And so by looking at these fossils in our fossil cabinet, I want you all to notice what era they derive from. What time in Earth's history would they have been made? What time did these relics, some who do have living relatives, like the ginkgo, even if there's one, and some who don't, like the Lepidodendrales. But boy, what I wouldn't give to see the jungles, the swamp jungles of the Carboniferous with these really, really unique looking trees. After that, y'all will be doing some Wikipediaing on some of our living fossils. So go through the exercises, look up what a hagfish is, and take a look at some of those ancestral characteristics of jawless fish that the hagfish have. Same with our crocodiles and some of our other plant species. I'd also encourage y'all, as part of the homework for the remainder of this class, watch a couple of nature documentaries. Watch some things about species that have lived before, how life has evolved and changed on planet Earth. I think this is really a fun exercise to do for the remainder of the class, and watching those documentaries will help connect the things that you're learning in class to artistically made movies and the like. After all, a lot of Jurassic Park is based on fossils as well, so you can make cool movies from that stuff. That said, do some more reading, hang out in class and take a look at some of these fossils, and I'll see y'all next week.